हेलो भाई। कैसे हैं आप लोग? आई होप यू आर डूइंग गुड. सो वी शुड कंटिन्यू अवर discussion on calcium. Uh, the subject is the role of calcium in xenobiotic toxicity. And I told you in my last lecture that calcium is not redox active. Calcium is an essential element. Calcium is required by the body for different biochemical, physiological functions. The main depot of calcium in the body are bones. 99% of calcium is in the bones. Then the nails, then the hair, then the teeth. All these hard structures, they contain the higher amount of calcium. But the soft tissues like liver, kidney, lungs, their cells, they have a concentration of calcium up to the level of 100 nanomoles. 100 nanomoles is the concentration. It's a very small concentration. So the calcium regulation is performed by the help of hormones, and the major hormone that plays a role is parathormone, and the calcitonin that is secreted by the thyroid gland. So there is a feedback mechanism. Whenever the cell requires the calcium supply is maintained through that feedback mechanism. But what happens when a xenobiotic, like tributylene, like carbon tetrachloride, like paraquat, like acetaminophen, that is paracetamol, that enters into the system, and through circulation it reaches the cell, say for example the liver cell. Then what will happen? This will lead to some toxicological consequences. And before understanding those toxicological consequences of higher concentration of intracellular calcium, we will have to understand that the calcium entry into the cell is a very well regulated mechanism. The plasma membrane does not allow excess of the calcium to enter into the cell. It doesn't allow calcium to enter into the cell. There is a calcium pump, well established calcium pump, that maintains the 100 nanomoles of calcium concentration, intracellular concentration. There are two concentrations, the calcium which is present in the extracellular fluid and the calcium which is present in the intracellular fluid. And there is a gateway. The extracellular cellular calcium concentration is higher and the intracellular calcium concentration is lower. So a gradient is maintained. A gradient is maintained, and whenever calcium is needed, through that gradient mechanism, the calcium is allowed to enter into the cell. And it plays an important role for many of the physiological and biochemical reactions. Say, for example, calcium is essential for ATPase. Say, for example, it is a sodium dependent ATPase, the calcium dependent ATPase, the potassium dependent ATPase. We have got these ATPases like this. And the calcium is a catalyst for many of the phospholipases. Calcium works as a catalyst for many other enzymes. So calcium does play a very important role. And calcium is not redox active, it is not causing oxidative stress, it is not playing a very... But these xenobiotics which induce the oxidative stress, that may lead to calcium perturbations. You understand it? Calcium itself does not cause any damage, but the chemicals which induce oxidative stress, these oxidative stress conditions, ER stress conditions, nitrosative stress conditions, they lead to disturbances in calcium concentration inside the cell, outside the cell, or calcium homeostasis is disturbed. That we are going to discuss. And for that, we are to discuss what will happen if the amount of calcium increases in the cell. First question: what will happen if the amount of the calcium exceeds, intracellular calcium exceeds in the cell? It will it will be there in the cytoplasm. It will be through transporters, it will be going to mitochondria, it will be going to the endoplasmic reticulum, it will be going to the even to the nucleic acids, it will be going to other organelle of the cell. And wherever the mechanism of its sequestration occurs, that will do. But if that also overwhelmed, that is also overpowered, then what will happen? This is the main purpose of today's lecture, and these are called as the toxicological consequences of higher amounts of intracellular calcium. Now, number one, what will do? What calcium will do? Number one is that calcium, when excess calcium will enter into the cell, it will activate certain enzymes, and these enzymes are called as caspases. These enzymes are called as caspases. So the first thing that this intracellular calcium is going to do is to increase the activity of caspases, and these caspases are lytic in nature, and these caspases will lead to the the activation of, or the introduction of, or induction of apoptosis. We call them as executioners of apoptosis. Caspases are the executioners of apoptosis. एपोप्टोसिस को एग्जीक्यूट करने वाले अगर कोई प्रोटीन या एंजाइम है तो उनको हम कैस्पेज कहते हैं. तो पहला काम इंट्रासेलुलर कैल्शियम हायर कंसंट्रेशन करेगी सेल के अंदर कि कैस्पेज को एक्टिवेट कर देगी, और कैस्पेज एपोप्टोसिस कर देंगे, न्यूक्लियर फ्रेगमेंटेशन कर देंगे, प्रोटीन डीनेचुरेशन कर देंगे, एंड सेल विल मूव टुवर्ड्स प्रोग्राम्ड सेल डेथ. Second thing that these caspases, the higher amount of intracellular calcium will do, that it will activate certain other proteins, and these proteins are called as calpains. I told you in my last lecture also. These calpains are they sequester the calcium, but their excess formation of these calpains will lead to the deposition of, will lead to the what you call as the cytoskeleton. They will be affecting the cytoskeletal disruption. I told you, the microtubules is cytoskeleton. You must have studied the structure of a cilium or flagella, and that structure shows some cytoskeletal components. The cell is bounded by the cytoskeleton filaments, but you don't see them. You cannot observe them. They are cadarines, they are spectrines, they are vimetine, and like this we classify them. Actin and fibrin and myosin, these things, they are encircling the outer side of the cell next to the plasma membrane, the cell membrane. What will happen? These calpains will disturb the cytoskeletal components. Nature has done something wonderful. To antagonize the effect of calpains, nature has given the cell another protein which is known as calplastin. You should know that. Calpain, calplastin, calmoludin, calcitonin, ये सब कैल्शियम डिपेंडेंट मॉलिक्यूल्स हैं. तो कैलप्लास्टिन क्या करेगा? कैल्पेन को न्यूट्रल करने की कोशिश करेगा कि भाई एक्सेस ऑफ द कैल्पेन जो तुम बन रहे हो, तुम्हारा सिंथेसिस हो रहा है हायर अमाउंट ऑफ कैल्शियम की वजह से, कहीं तुम साइटोस्केलेटल डिस्टर्बेंसेस ना कर दो. I told you that the blabbing of the cell occurs due to cytoskeletal disturbances. कहीं तुम साइटोस्केलेटल डिजॉल्व ना कर दो और सेल नेक्रोसिस की तरफ ना चलने लगे, तो इसलिए नेचर ने क्या दिया? कैलप्लास्टिन. कैलप्लास्टिन उसको एंटागोनाइज करता है, उसको दबाने की कोशिश करता है, लेकिन जब वो ज़्यादा हो जाते हैं तो कैलप्लास्टिन भी मजबूर हो जाता है और जब वो मजबूर हो जाता है तो फिर ये कैल्पेन क्या करते हैं, दे रिजल्ट इनटू द डिसोल्यूशन डिसरप्शन ऑफ द साइटोस्केलेटल कंपोनेंट्स एंड द रिजल्ट ऑफ द, वी कैन कॉल द टॉक्सिकोलॉजिकल कॉन्सिक्वेंस ऑफ हायर अमाउंट ऑफ कैल्शियम इज number one, activation of specific enzymes known as caspases. Number two, the toxicological consequence of higher amount of calcium is the activation of calpains that leads to the higher synthesis, or that leads to the disruption of these cytoskeletal components, and it finally leads to necrosis. अब थर्ड पे आते हैं कि हायर अमाउंट ऑफ, और ये ऐसा नहीं कि एक एक करके हो रहा है. इट्स नॉट लाइक दिस दैट भाई पहले जो है वो कैस्पेज एग्जीक्यूट कर लो, फिर जो है कैल्पेन कर लो, फिर कोई और काम कर लो. नो, इट्स अकरिंग साइमल्टेनियसली. देयर इज ए क्रॉस टॉक. Cross talk is, I like this word very much. That in cell there is a cross talk. Cell and cell, they do cross talk. So there is a cross talk amongst them. So next thing is that higher amount of calcium will activate certain enzymes which are known as endonucleases. क्या कहते हैं उनको? Endonucleases. और ये endonucleases क्या करेंगे? These endonucleases will will break DNA strands. इस DNA के साथ जा करके जो DNA स्ट्रैंड है उसको ब्रेक कर देंगे. एंडोन्यूक्लियसेस DNA स्ट्रैंड ब्रेक करता है, इसीलिए उनका नाम हमने पढ़ा है, उनका नाम करा है एंडोन्यूक्लियसेस. they will cause DNA breakage. For the example for that is acetaminophen, that is paracetamol. When higher amount of paracetamol is given to some experimental animal, the intracellular calcium level goes up. And when we administer EGTA, which chelates calcium, the level of the intracellular calcium goes down. So it has been confirmed now that acetaminophen EPA toxicity or paracetamol toxicity or paracetamol induced necrosis is mediated through higher level of intracellular calcium, and that is through the activation of endonucleases which lead to the DNA breaks. That leads to disturbs the genomic integrity. ज़्यादा अच्छा है वर्ड लग रहा है. They have got a pleiotropic effect. Pleiotropic means बहुत सारे effect, and they lead to genomic loss of the genomic integrity. So this is how the higher amount of intracellular calcium is reacting with endonucleases. The next point we have discussed, number one, caspases, which is responsible for apoptosis. Number two, the cytoskeleton disturbances. Number three is the DNA breakage, and number four is the activation of phospholipases. I told you in the last one also that the membranes, they have got a very important set of enzymes which are known as phospholipases. And phospholipases, activation of phospholipases leads to the accumulation of phospholipids in the cell. These are calcium dependent enzymes, and they lead to the inactivation or inhibition of arachidonic acid in the membrane, and they disturb they lead to the deposition of the phospholipids in the cell, and these phospholipids, deposition of these phospholipids leads to membrane disturbances. There are certain evidence that indicate that higher amount of calcium in the cell leads to mitochondrial injury, and that mitochondrial injury is because phospholipase A2 resides in the inner mitochondrial membrane. So when it is activated, it leads to mitochondrial injury, and the mitochondrial injury leads to is expressed or is shown by the opening of the hypothetical mitochondrial permeability transition pore, MPTP. We shall be talking of MPTP in next lecture when we are going to discuss the energy disturbances which are caused by xenobiotics in the cell energetics, how the xenobiotics, they affect the cell energetics. So here, the fourth consequence of the higher amount of intracellular calcium is that they activate this category of enzymes, that is phospholipases. They lead to the inhibition of arachidonic acid, that leads to deposition of phospholipids, and that leads to the membrane damage, that leads to the mitochondrial damage, and ultimately the MPTP opens and the influx and influx of the materials is disturbed, homeostasis is lost, it leads to necrosis. So activation of caspases led to apoptosis. The activation of calpains led to apoptosis. Activation of endonucleases led to apoptosis necrosis. Activation of phospholipases led to the deposition of phospholipids and the mitochondrial disturbances. Some of the intracellular calcium, there is an intracellular calcium pool, and the mitochondria enjoys that pool, the endoplasmic reticulum enjoys that pool, and when excess of the calcium is there, sometimes it goes to the lumen of the endoplasmic reticulum. Endoplasmic reticulum has got such type of structure, and there is a lumen, so calcium enters into the lumen of endoplasmic reticulum. And when it enters into the lumen of endoplasmic reticulum, it causes endoplasmic reticulum stress. You might have not studied it so far. Maybe sometimes we will be discussing it in other terms. And when this calcium enters into mitochondria, this is another very important consequence, that there is a reciprocal arrangement between calcium and sodium, and that is maintained by a molecule which we call, by a protein which we call as NAD, nicotine adenine dinucleotide. And when this NAD decreases, that leads to the depletion of ATP, that leads to the collapse of the mitochondrial membrane potential, because ATP is maintained by the mitochondrial membrane, and the excess of the calcium, it antagonizes sodium, and when it antagonizes sodium, there is the decrease in the amount of NAD in the mitochondria, and the depletion of ATP occurs, and it leads to mitochondrial membrane potential, collapse of the mitochondrial membrane potential. So the mitochondria is affected, nucleic acids are affected, DNA is affected, specifically the endoplasmic reticulum is affected, the plasma membrane is affected. So higher amount of calcium is going to affect each and every organelle of the cell. Then the next is the gene expression. Effect of higher amount of intracellular calcium will be disturbing the expression of the gene, especially the C, Jun, Miss, and the FOS, C-FOS. These genes are involved or are controlling the cellular death. They are controlling they are known as the cell death receptor pathways. These genes are very important in the cell death, because these genes are working in cell disruption pathways. So next consequences. I have discussed with you 5 consequences. The first consequences was the activation of caspases, second was activation of calpains, third was activation of endonucleases, fourth was activation of phospholipases, fifth is the gene expression changes, and there was one more, that it affects the mitochondrial NAD, and next is there is a crosstalk. ये 5 चीज़ जो है इनके बीच में क्रॉस टॉक चलती है. कैल्पेन कैस्पेज रिलेटेड है, कैस्पेज एंडोन्यूक्लियस से रिलेटेड है, एंडोन्यूक्लियस फॉस्फोलिपेज से रिलेटेड है, ईआर माइट्रोकॉन्ड्रिया रिलेटेड है, माइट्रोकॉन्ड्रिया ईआर रिलेटेड है. आप एक को छेड़ दो, सारी चीज़ें छिड़ जाती हैं. आई ऑफन टॉक टू यू. It's like a mosquito net. If you disturb one knot, the entire cell, the homeostasis of entire cell is lost. Then the calcium is a important mediator of cell death. बहुत लोग काम करते हैं अफेक्ट ऑफ कैल्शियम ऑन सेल डेथ, स्पेशली ऑन एसिटामिनोफेन, कार्बन टेट्राक्लोराइड पर पहले करते थे, अब नहीं करते। अब स्टीन उरेनियस फ्रॉम कैरोलिनस्का इंस्टीट्यूट इन स्वीडन इज स्टिल वर्किंग ऑन एसिटामिन toxicity, is still talking of calcium. There is another gentleman, Kapler, D. Kapler. He has also worked on the role of calcium in cell death. There is a book by Urs Borstelli from Italy. It has a chapter on disruption of intracellular calcium homeostasis and the toxicological consequences. So the calcium, इंट्रासेलुलर कैल्शियम में डिस्टरबेंस क्यों होगा? ऑक्सीडेटिव स्ट्रेस की वजह से, खुद नहीं करेगा। क्यों होगा? रेडिकल्स की वजह से। ये क्या करेंगे? ये इंट्रासेलुलर कैल्शियम के लेवल को बढ़ा देंगे और वो फिर ये 5 कैटेगरी के जो अफेक्ट मैंने आपको बताए, इन 5 कैटेगरी के अफेक्ट्स को मैनिफेस्ट करेगा, इनको क्रियान्वित करेगा। इस तरह से सेल क्या होगी? या तो एपोप्टोसिस से मरेगी या नेक्रोसिस से मरेगी। एपोप्टोसिस क्या है, नेक्रोसिस क्या है, इसकी चर्चा हम फिर से अलग से करने वाले हैं, अगले लेक्चर्स में करने वाले हैं, बिकॉज दैट इज़ द मेन थिंग दैट वेयर वी आर गोइंग लीड टू. So I hope the things must be clear to you now. Enjoy studying. We shall meet again.